What W wanted me to talk about today was a couple areas we've been doing research in lately. And one of those areas is really what's going on in the rumen relative to trace mineral source and how that may influence fermentation. And then this other part of this presentation really is a few thoughts and a study that we conducted about zinc and chromium supplementation in feedlot cattle. So that's in general the two topics I want to discuss today.
There, you can say that again, sorry.
We okay?
Can you say that again, something? We miss you.
W, we okay?
Don't think so. Can you hear him? Can you guys hear him? But just in the head, vocês só podem ouvir no headset, né? Eu tô ouvindo ele agora.
No, still.
Oh, okay. Go ahead, Kenny, please.
Okay, yeah. So just in general, I put a little outline together talking about the background associated with our interest in understanding the rumen fermentation and how minerals may influence rumen fermentation. And so I want to talk a little bit about copper and zinc and the microbial function, some solubility studies that were done in the early 2000s with the IntelliBond copper and zinc, and then spend a little bit of time talking about a particular project that was done out of Bill Weiss's lab at Ohio State University, looking at the IntelliBond trace minerals and how they influence fiber digestion. And then I'll jump into or move into our studies with rumen solubility and how trace mineral source might be affecting rumen fermentation and the binding strength associated with the digesta. And then I'll spend a little bit of time talking about the effects of chromium and zinc on feedlot cattle growth. So I think it's important that we have a little brief conversation about the role that copper and zinc play in microbial fermentation. And what I tried to do is go back in the literature and look to see what has been done. And the majority of research that is looked at really the microbial requirements of copper and zinc, and to some extent manganese. Most of those, if not a large proportion of those studies, have been done in vitro. And so I went back in the early literature, and bottom line, I think, to summarize this, is that some of the earlier work and rumens using in vitro rumen simulated experiments really do indicate that the microbial copper and zinc requirement is relatively low, and so much lower than it would be from a dietary requirement for the animal. And so what I tried to do in this last bullet point here is really summarize from the literature what I think the copper and zinc requirements are for bacteria in the rumen. And this has not been published. This is just a summation of what I found in the literature. And what I come up with is about 0.1 milligrams of copper per liter and about 0.2 milligrams of zinc per liter will meet the microbial requirement based off of the response of dry matter digestibility. And so I think the important part is these are several orders of magnitude lower than what the dietary requirements are for ruminants. And I also pulled this older paper out. This was published by Emanuel and Staples in 1990, where they were looking at ruminal disappearance of copper and zinc from forages, different forages, using the in situ approach. And what really stood out to me when I looked at these data, and here's the bunch of different forages here on the left. And at this zero time, this is the percentage of copper that was released. So this is basically soluble in water. So the copper associated with the alfalfa, Bermuda grass, any of these forages here was highly soluble at time zero. And then 72 hours later, we see an increase due to digestibility, most likely, of the copper release from these forages. Same goes with zinc. It's a little bit less soluble at time 0, but it does become very soluble in 72 hours. And really what this makes me think about is that when an animal consumes a forage, they're probably at some point in time, probably pretty quickly after that forage hits the rumen, there's probably microenvironments with very, very high copper concentrations until the dilution of that mineral can take place in the rumen. And so I thought this was pretty interesting that the majority of copper, at least in these forages, is highly soluble in just water at time 0. So what's that mean for the bacteria in the rumen? And there has been a lot of research work conducted in trying to understand metal homeostasis in bacteria. We do know I just summarized these bullet points here from a regulated metal. The majority of bacteria have ways to regulate uptake of and or reduce the uptake of metals. They can pump metals back out if they do get into the cell, and they also have binding proteins that they can help to prevent any further action from metals if they do get into the bacterial cell. But when I start look at all the data, what I would say is that if you feed a practical diet, a pretty standard type diet to beef cattle, without supplementing any copper and zinc supplementation, I do believe, and the data would indicate, that you are able to meet the microbial copper and zinc requirements. I'm just focused on copper and zinc here. But that's way different than the animal's requirements. And I would say that we do need to remember that there are a lot of factors that can impact the solubility of minerals, and we've spent a lot of time trying to understand how rumen pH or the concentration of dietary antagonists may influence the solubility of copper and zinc primarily, diet type, which I'll have a little bit of data on here in a minute, and then mineral source. And so this really got us into the area of what is happening if a mineral, in this case, copper and zinc, minerals are highly soluble in the rumen. Does that inhibit or somehow affect fiber digestion? So in the early 2000s and then a little bit later, there was some work done out of Dr. Spears's lab where they were looking at the solubility of the hydroxy copper sources and zinc sources. And just to summarize this particular slide, in general, copper hydroxychloride and zinc hydroxychloride are pretty insoluble in water at a pH of 7. If you were to supplement copper sulfate and or zinc sulfate and put it in water, the majority, over 80 to 90% of the metal, copper and or zinc, will become ionized or soluble at a pH of 7. Now, if you reduce the pH to what you would typically expect to see in the abomasum, perhaps in a ruminant, down to 2 or 3, the copper and zinc from the hydroxychloride sources does become highly soluble. And so we thought this was going to be an interesting approach because you have potentially minerals sources that are not soluble in the rumen that may be soluble later on in the digestive tract. So that's kind of what led us down this road of trying to understand the impact of trace mineral source solubility in the rumen and on essentially fiber digestion. This is the paper I mentioned earlier out of Bill Weiss's lab at Ohio State University, and they did a really neat study where they had two treatments, a sulfate treatment and a hydroxy treatment. And the copper, zinc, and manganese, you can see here, we're at 10, 30, a little over 30, and 30 parts per million supplemented in the diet. And what they did in these lactating dairy cows is they looked at NDF digestion. And they had two different types. They have a high forage-based diet and then a byproduct-based diet. And regardless of the type of forage or byproduct that they added to the diet, you see that there's about a 2% increase in NDF digestion associated with the animals that were consuming hydroxy trace minerals. And they made a statement in their paper that was, I think, pretty unique, that they made this statement that indicated that potentially the solubility of the high solubility of the sulfate trace minerals in the rumen may be impacting fiber digestion and or causing the bacteria to have to expend more energy to either bind or pump or remove the metals, the soluble metals out of their cell or away from them. And so that's what led us on to a series of experiments. And so what I wanted to do, my first experiment with the graduate students here was to see if we saw the same type of change in fiber digestibility that the folks at the Ohio State University did in dairy cows. So we were interested in the beef cattle side of things, and so we did a really quick study of 8 crossbred steers. We had, fitted with ruminal cannulas. We fed these steers corn, silage, steam flake corn-based diet. So on a dry matter basis, that was about 50% corn silage, 50% steam flake corn. And the treatments were really straightforward. There were sulfate and hydroxy trace minerals, so 10 parts per million copper, 20 manganese, and 30 parts per million zinc. And so we just did a real quick digestibility study, and then followed by a 5-day fecal and urine collection study. So the results came out, and it still intrigued me. We did hold these animals' dry matter intakes constant, so there's no difference there. We did not see a difference in dry matter digestibility, but when we narrowed down and looked at the NDF digestibility, we saw a tendency anyway for roughly about a 2 or 3% increase in NDF digestibility. So this is one of my former graduate students put this together, and so it got us thinking about, okay, what is really going on in the rumen with these different sources of trace minerals? And if you understand that those hydroxy trace minerals are relatively insoluble in the rumen, and the sulfate trace minerals typically are soluble in the rumen, what's really going on? And so that's what led us to a series of experiments with the primary objective of basically determining the impact of these trace mineral sources, hydroxy trace minerals and or sulfate trace minerals, on dry matter and fiber digestibility. Also, the soluble concentrations of copper and manganese and zinc in the rumen. What did that do to fermentation characteristics such as volatile fatty acid production? And then the thought process was that if it's not soluble, if the hydroxy trace minerals are not soluble, they have to go somewhere. And so where did they go? And so what we wanted to do is try to understand if those minerals, if the hydroxy trace minerals were associated with the solid digesta, how tightly were they bound to the solid digesta? And so we used a kind of an older technique to try to parse that out or figure that out. So again, a lot of similar types of designs here. We had a few more steers with cannulas, did a very standard what I would call a standard digestibility study with collection of feces and urine. This first group of steers was fed a low quality grass hay based diet. We did supplement a little protein to it, but the treatments were 20 parts per million copper, 40 parts per million manganese, and 60 parts per million zinc from either sulfate or hydroxy sources of trace minerals. And again, not a really complicated experiment, just a lot of work. Here's the diet, just so you can see that the acid detergent fiber NDF is relatively high, and we did supplement protein to increase the protein content of the diet to about a little over 12%. And this timeline will be very similar because we did this same study with a dairy type diet. So the first 7 days of the experiment were at diet adaptation, where we fed the animals their treatment source, the diet, forage-based diet plus their treatments. We brought them into the metabolism barn. We did a 5-day dry matter determination. experiment so that we knew what these animals were eating, so that we could pair feed these animals during the further adaptation period, so that we limited their feed intake to be equal within a pair. And then we did a real simple total fecal and urine collection study. And so just to get into the data real quick, again, like I said, we held dry matter intake constant so that we wouldn't have any differences in dry matter intake. We saw a tendency for dry matter digestibility to be higher in the animals receiving hydroxy trace minerals, and then ADF and NDF digestibility were significantly higher in the hydroxy supplemented animals, and a tendency for crude protein digestibility to be higher. So this is relatively new information, and we wanted to figure out, okay, really what is going on here. And so on day 6, so after we finished the fecal and urine collection study, we took rumen fluid samples 0 and 2 at times 0, 2, and 4 hours post feeding. And what we're trying to understand is what is going on with fermentation kinetics, if you will. And really what we saw is no effect on rumen pH. Butyric acid was lower in the hydroxy trace mineral supplemented animals, and then total short chain fatty acids or volatile fatty acids was higher in the hydroxy trace minerals. So that led us to be thinking, well, maybe some of these trace mineral sources that are highly soluble in the rumen may be impairing fermentation. So to try to understand more about the solubility aspect, what we did after we did the VFA analysis on those animals is we removed them from their dietary treatments for 14 days. And then we brought it back into the digestion stalls and just really essentially gave them a bolus dose of the copper, manganese, and zinc at 2 times the National Academy of Science, Engineering, and Medicine requirements. So we either gave them 20, 40, or 60 parts per million of copper, manganese, and zinc respectively from either source. And then the real fun part for this project was every 2 hours. So we gave them the bolus dose, we mixed it in by hand into the rumen, and then for every 2 hours, we took samples through 24 hours. And so we started at -4 and -2 and then 0, and we dosed them, and then over 24-hour period, we took samples. And so the next series of graphs will kind of show you what the solubility aspect was in this particular experiment. So on the y-axis here is the soluble copper concentrations in the rumen, and the black line is the sulfate trace minerals, and this is copper sulfate, and then this would be the hydroxy copper source. And you can see that at about 2 or 4 hours or so post bolus dose, we get a pretty large spike in soluble copper concentrations from the animals receiving the copper sulfate. Whereas the hydroxy trace minerals stayed pretty flat throughout the solubility of the hydroxy trace minerals stayed pretty flat throughout the 24-hour period. We also looked at zinc, and we see a similar type of pattern, I guess, if you will. And by 2 or 4 hours post bolus dosing, you see this that becomes different. So the hydroxy zinc source has a lower solubility at about 2 to 4 hours post bolus dose, all the way through about 12 hours. And so that was pretty interesting to us, and it was making some sense, but we still wanted to figure out, okay, where's the zinc going from the hydroxy trace mineral source or the copper if it's not becoming soluble. Now, one caveat to this, like any other experiment, is that the manganese side of things was way different. And it gets more interesting later, but it appears to me that the manganese solubility and the sulfate versus hydroxy are pretty similar. We weren't expecting this, but these are the numbers we got. So we're still kind of scratching our head about this data set, but we're going to continue to investigate why these were pretty similar. Okay, so to try to understand the binding strength, so if the copper from copper hydroxychloride is not in solution, where is it? And so what we did from those grab samples we took every 2 hours is we removed the liquid fraction and we kept the solid fraction, or the indigested or undigested material. And so we wanted to estimate a release of copper, manganese, and zinc from the solid digesta. And so we used a really old technique, but it's pretty interesting and a very reliable type of approach. But it was really through dialysis. And so what we did, and this is just a kind of a picture of what we did. So we took these dialysis bags and we put our digesta inside the bag. And then we have in this solvent here is a strong chelator. So anything that's loosely bound to the digesta material will be moved through the bag and into this portion of this beaker. And so you let it equilibrate for about 12 hours, and it has to be in a bunch of different parameters. But long story short is what we're trying to do is if these are not tightly bound to the digesta, how much is actually coming out. And so that would be a calculation or an approximation of the digestibility, or the releasability is probably a better way to put it. So here's just one data set. Obviously, this is the 12-hour mark. So we took the digesta from the samples we took at 12 hours. We've done many more, but this is about where we saw the most release, and at least for copper. And so here's the initial concentrations in the digesta. And so the hydroxy trace minerals, so copper was much higher in the digesta than it was from the hydroxy trace mineral sources than the sulfate. And you can see that manganese about the same, and zinc was much higher in the digesta fraction at 12 hours post bolus dosing. And then so we ran dialysis. So we dialyzed the material against CDTA, and we get about a 60% release of copper in the hydroxy supplemented animals, and about a 26% increase in the sulfate or release. Manganese was just the opposite of what we understood, so we still got some questions about manganese. But if you look at zinc, about 88% of the zinc that was in the digesta was able to be released from the digesta. So that tells us that it's not bound in a way that will influence the digestion or absorption later on down the GI tract. And so real quick, I don't want to run over, but we did the same study. So I'm going to kind of go through this a little quick. We did the same study with dairy type diet, and here's the diet. So it's pretty heavy in corn silage and alfalfa hay and some distillers grains, but we also added another treatment. So we had the sulfates, we had the IntelliBond, and then we had the organic trace minerals. And again, same general experimental procedures. And you can see here that dry matter intake, we held that constant for this particular experiment too. Dry matter digestibility was not different. NDF digestibility was greater in the organic and the hydroxy trace mineral supplemented animals, as ADF was as well. And then, and we didn't see an effect on crude protein digestibility. And so we did see this similar response to hydroxy trace minerals, and then the organic trace minerals were similar to the hydroxy trace minerals in this particular experiment. Now, it's much different, though, when you start to look at the solubility aspect in the rumen. So again, we took VFA rumen samples for VFA analysis, and we did see that the total VFAs were greater in organic and hydroxy animals receiving organic trace minerals and hydroxy trace minerals compared to the sulfate trace minerals. But when you look at the solubility aspect, hydroxy trace minerals behave the soluble copper concentration is really similar to what we saw in the first experiment when we had the bolus dose. So we gave these animals the same bolus dose with their treatments. The organic was very soluble. The copper from the organic was very soluble, and the copper from the sulfur or copper sulfate was highly soluble. And the thought process here on why the high solubility of the organic trace mineral may not be influencing fiber digestion or digestibility in general is that it may still be chelated to its whatever ligand that it was bound to. And so we see a similar response to zinc here as well. And then manganese was all over the board again. We're not really for sure. There's not a lot of differences in manganese solubility between all these sources. So just to give a brief conclusion here, we do think that rumen solubility of copper and zinc differs between the sulfate, the organic, and the hydroxy sources. And it appears that I think if we have rumen copper and zinc solubility may impact rumen fermentation in forage and dairy-fed type diets. And we are completing another study. We've completed the animal work, but where we're looking at a very, very high concentrator or feedlot type diet, same general procedures. And so hopefully we'll be able to get that published later this year. Kind of shifting gears, Davi wanted me to talk about one particular study we've published regarding chromium and zinc supplementation, and so why the interest in chromium and zinc? Well, if you look through that, I'm not going to read all of these, but zinc is heavily involved in growth and development, and it's involved in many enzymes in the body and in various metabolic pathways. We do know that if you remove zinc from the diet and the animals become zinc deficient, that growth is impaired, the immune system is impaired. And so from a recommendation standpoint, the NRCs of the world or the nutrient requirements for beef cattle would suggest that it's about 30 parts per million. However, if you look at the surveys that are done or the consulting nutritionists, at least in the US, these high zinc concentrations in diet, they would, on average, the consulting nutritionist would supplement 90 parts per million of zinc per kilogram of dry matter into the feedlot diets on top of whatever's in the basal ingredients. And so this is much different than what's actually recommended, but what in practice, these high levels of zinc are pretty common. And I think if you look back in the literature, I just pulled one of these papers out, but there is so increasing zinc in the diet above the NRC requirement in certain situations does improve average daily gain and feed efficiency and did alter marbling scores. And this, Dr. Spears in this was looking at different zinc sources, but in general, those high levels of zinc in this particular study was about 60 parts per million total diet. You did see an improvement in growth and marbling. And so the combination of chromium, so why would chromium be of interest, is that chromium has been shown to enhance function of insulin, so that it's having an impact on maybe lipid metabolism, carbohydrate metabolism. The challenge with working with chromium is that there's contamination all over the place. If you run a feedstuff through an auger that's metal, you're going to get contamination. This study right here fed a really, really high dose of chromium, and they saw improvements in dressing percentage and marbling scores. Other studies, though, I just want to caution everybody that you can find in literature that other studies show no effect of chromium supplementation on performance, on growth performance and or carcass characteristics. The challenge with that, because of the variability in results from one study to another, is that it was really difficult for the National Academy of Science, Engineering, and Medicine and Engineering really struggled with trying to determine what the requirement was for chromium for beef cattle. And there just was not enough information, and some of it was conflicting, the published information. And the challenge with determining a requirement is you need to be able to make an animal deficient and then supplement up to a certain level to where those deficiency signs go away. And we just haven't seen any of that information published yet. And so wasn't able to come up with that group was not able to come up with a requirement for chromium. But what we did, I was interested in this, and so we real quickly we had 400 steers, 8 blocks within this 8 weight blocks, 5 different treatments. So we had a total of 40 pens with 5 different treatments, 10 animals per pen, and we fed them a finishing diet for a little over 180 days. So here's the finishing diet. It's a little over 13% crude protein, 1.44 megacalories per kilo, and the NDF was pretty low at 12.5 or a little over 12.5%. Here's our treatments. 90%, and this is 90 parts per million on top of whatever was in the diet with chromium. So it's at 0.25 parts per million or 250 parts per billion. We fed in sulfate. We had an IntelliBond source with chromium, a zinc source with chromium added, and then we had the IntelliBond source at 90 with no chromium. And then we had a 90 milligrams of zinc from a blend of zinc sulfate and or zinc methionine, and then we had a lower dose of IntelliBond Z with chromium. And so I'm going to go through these numbers really quick, but in general, what we saw is that final body weights were roughly, we did not see any zinc effects here, but with the high zinc at 90 parts per million, if chromium was added, we did see roughly a 12 kilo improvement in final body weights with about 1/10 of a kilogram of average daily gain improvement. So that was suggesting that chromium may be influencing not growth performance. When we look at hot carcass weights, we did see that animals receiving supplemental chromium at these high doses of zinc had roughly about a 7 kilogram increase in hot carcass weight. And we also did see that chromium did influence a longissimus muscle area by making it a little bit larger. And so when we summarize these data, really it is, we didn't see a zinc source effect, but I think overall these data would indicate that zinc and chromium supplementation to feedlot cattle may improve growth performance and carcass characteristics. And so our next big push is to try to understand the mechanisms by which zinc and chromium may be doing this.